Hi, am I audible?
Yes, ma'am.
Okay. So, we'll start the session. So, today is the 10th problem solving session for the course Environmental Chemistry and Microbiology. The course instructors are Professor Anjali Pal and Professor Sudha Goyal from IIT Kharagpur, and I am Shrija Ghosh, a PMRF from IIT Kanpur. Uh, so, today we'll discuss certain questions related to your 10th week's topic, that is microbial growth and kinetics. So we'll discuss the questions that were related to your week 10's assignment. Feel free to ask any doubt if you have. Just let me know whenever you have any doubt. I'll be asking certain questions. Try to answer those questions and then we can discuss if the questions are right or wrong so that you can have a better clarity. Okay, so starting with the first question. Question number one. The process in which all living cells, spores, and viruses are completely destroyed from an object is called option A, disinfection, option B, pasteurization, option C, sterilization, and option D, antisepsis. The process in which all living cells, spores, and viruses are completely destroyed from an object is called disinfection, pasteurization, sterilization, or antisepsis. What do you think is the answer? You can write the answer in the chat box and then we can discuss. Option A, disinfection. Option B, pasteurization. Option C, sterilization. And option D, antisepsis. Can you answer this question? You can write the answer in the chat box. Just try to take a guess even if you don't know the answer. Yes, so you have written option C, that is sterilization. That is correct answer. That is the correct answer. Option C, sterilization. So, these all these options are different methods of removing microorganisms. So, disinfection is a process where destruction of vegetative pathogens uh, is done by using physical or chemical methods such as chlorination, ozonation. So, just a second. Please mute your mics, all of you. Okay. Okay, so a lot of people joined, so I'll start with the first question again. So the first question was the process in which all living cells, spores, and viruses are completely destroyed from an object is called. So the correct answer is sterilization. So I was discussing what are the other processes. So all these processes are basically uh, used to remove microbial microorganisms the from an object or from a specific solution. So there are different techniques, and every technique has a meaning, and every technique has a purpose. So, starting with disinfection. So, the simplest example that I can give is when you use the floor cleaning solutions when you are mopping uh, at your home. When you mop the floor, you use certain cleaning solution like Lysol and all. So those are disinfecting solutions that is done to remove any vegetative pathogens that are present. Uh, to remove those pathogens from the floor, we use the disinfectant. Same thing, like the other examples are chlorination or ozonation. So all these techniques, they can help removing the vegetative pathogen. Okay, destroy the vegetative pathogen. Now comes the second option. Now comes the second option, that is pasteurization. So this is most commonly used in the milk industry, in the dairy industry. So you must have seen whoever uses milk packets, the packaged milk that we get from any of the companies. There are so many dairy companies, right? So when you see on the packets, the like milk we get in the packets, it's written pasteurized milk. Okay, so what do we mean by pasteurized milk? Pasteurization is a process where we uh, heat the milk at a very high temperature for a very short duration to kill all the pathogenic microbes. Okay, so it's a partial sterilization process. We are not killing all the microbes, we are just killing the pathogenic ones so that we don't get sick when we are drinking the milk. Okay, so that is a uh, partial sterilization process, and we use pasteurization in other industries as well, other beverage industry, other food industry as well. But milk is a very common example. So whatever milk you are getting in the packets, all of them are pasteurized milk. Okay, so that is pasteurization. Then comes sterilization, which was the answer for this question. So it is a process of removing all the living cells, spores, and viruses completely, okay, from an object. So if let's say I have a solution and I want to make the solution sterile. So what do we mean by sterile? Sterile means it doesn't contain any living cells or spores. Okay, so nothing is there, nothing living is there. Okay, or nothing, no, none of the spores are there that can lead to a living cell. So no organism is present, okay, in the solution. So that is called a sterile solution. Now, to get that sterile solution, we do a process called sterilization where we remove all these uh, spores and bacterial cells and all. Okay, so sterilization can be achieved by many techniques. So autoclaving is one of the most common technique that we use. So what we do is like as I gave the example of a solution. So if I want the solution to be sterile, what will I do? I'll put it in an autoclave. An autoclave is like a big pressure cooker. Okay, you can understand like that. So it is a big pressure cooker and pressure gauge is attached to it. So when we heat, like in at your home, you must have seen uh, uh you must have seen that we use pressure cookers for cooking rice, right? Right. So what do? We add the rice, we add water, and then we heat the cooker, and after certain point, at a particular temperature, the pressure is achieved, and at that time the whistle starts, because that is the maximum pressure it can withstand. After that, the whistle starts blowing, and the pressure is released from the autoclave, so from the cooker. So same thing happens in the autoclave. So autoclave also is calibrated in such a way that when we heat the water inside it, uh, it will reach and we are doing the heating under pressure like we are doing in pressure cooker. Okay, so same thing, same, we attach a rubber gasket and we seal it completely with a whistle kind of a structure, so that there's no way the vessel is completely sealed basically. So once it is completely sealed, and when we are heating it, obviously pressure is going to develop, and at a very high pressure it can reach a temperature beyond the water's boiling point. So water's boiling point, as you all know, is 100, right? So when we are heating the water under pressure, it can actually reach up to 121 degrees Celsius, and that's a very high temperature. So at that temperature, a lot of living cells and microorganisms are destroyed, and that is a very common way of achieving sterilization. There's another way of achieving sterilization. Is membrane filtration. So what we do is all the smallest microorganism is generally considered as bacteria. Okay, so viruses are smaller than bacteria, but virus don't really grow without the host organism, so uh, it doesn't cause any great problems. So bacterial cells are generally larger than 0.22 microns. So we use a membrane filter, it's basically a filter paper which has a pore size of 0.2 microns, so that we can filter the solution whatever we want to sterilize. We filter the solution, and when the solution passes through the filter, all the living cells are stuck over the filter paper, and the solution becomes sterile. The solution that we are getting out of the filter paper is sterile. Okay, so that is one another technique. And the fourth option was antisepsis. So antisepsis is the destruction or inhibition of microorganisms on living tissue to prevent their growth by using antimicrobial agents. So antisepsis is basically usage of any antiseptic uh, agent. So the example I can give is when you get a wound. uh, let's say you hit yourself and you get a wound. Okay, so now first thing we generally do is put some Dettol or some Savlon, right? So those are antiseptic liquids. What do we do? uh, what So what do these antiseptic liquids do? They basically prevent or inhibit the growth of microorganisms on the wound. So now when you get a cut or when you get a wound, it's a open area, right? So if you get a cut on your skin, it's exposed to the environment. And when your skin is when your skin has a cut and just a second. Yeah. So when your skin has a cut and it uh, is exposed to the environment, a lot of okay, yeah. I was saying when your skin has a cut and when it is exposed to the environment, a lot of microorganisms can enter your body through that cut, right? So to prevent that from happening, we use antiseptic solution so that it can inhibit the microorganism growth on the living tissue. Okay. So these are the four types of techniques which are basically different. Like the goal is different, but all of the techniques are basically to remove certain microorganisms. Just give me a second. Sorry for the break. Yeah. So moving on to the next question now. Question number 2. What is a generation time? So option A is time required for the microbial population to generate energy in a culture. Option B is time required for the microbial population to double in number. Option C, time required for a microbial population to double in weight. And option D, time required for a microbial population to reach stationary phase. So what is generation time? Requesting all of you to write the answer in the chat box. What is the generation time? Time required for the microbial population to generate energy in a culture? Or is it time required for a microbial population to double in number? Or is it time required for a microbial population to double in weight? And fourth is time required for a microbial population to reach stationary phase. Okay, I have one answer, option B. Anyone else wants to answer? Requesting all of you to try to answer the question so that even if you answer it wrong, you will understand why your answer was wrong. Anyone else wants to answer? Write your answers in the chat box. No one else wants to answer? No one else knows the answer to this question. What is a generation time? What do you think a generation time could be? Take a guess. Okay, so I'll tell you the correct answer is option B. So you were right. Time required for a microbial population to double in number. So generation time is basically the time it takes for a population of bacteria to double in number. So a bacteria, when it divides by binary fission. So one cell basically splits into two. So first, it duplicates its genome. So the DNA is duplicated. So that is the replication part where two copies of DNA is present. So this is the mother cell, right? So the mother cell had a DNA, so it will make a copy of the DNA, exact copy of the DNA. So now it will have two DNA, and then the cell will start elongating, and later it will divide from middle. So there will be two daughter cell. One of them will have the parent DNA and one of them will have the new DNA. Then again, this daughter cell will divide into two, and then this daughter cell will divide into two, and like that the cycle will go on. Okay, so this is how binary fission occurs. So generation time is basically the time taken for the population of bacteria to double in number. So now if there's one bacteria, it will become two. Now imagine there are 1 million bacteria. So from 1 million, they will become 2 million. From 2 million, they will become 4 million. So the population will double in number. So the time required for this doubling. So there are certain bacteria that can require a few minutes to double, like E. coli divides in about 20 minutes. Okay. So, and E. coli cell, it can double in number in approximately 20 minutes. Okay, but there are certain bacteria that takes hours to grow. Okay, so they can take up to 1 or 2 hours to double in number. So one cell uh, will take 1 or 2 hours to divide into two daughter cells. Okay, and there are certain bacteria that can take only 10 or 20 minutes. So one cell will divide into two daughter cell in 10 or 20 minutes. So, and since this binary fission is happening, so you can see every time the number will keep on doubling, right? So it will increase exponentially, and the total population, basically the number of bacteria in a population will be equal to 2 raised to the power of n. That is the n is the number of generations. So if we start with one bacteria, let's say one bacterium, so one bacterium will become two bacteria after first generation. So 2 raised to the power of 1, that is 2. Now 2 bacteria will become 4 bacteria after second generation. So 2 raised to the power of 2, that is 4. Now 4 bacteria will become 8 bacteria. So 2 raised to the power of 3. So after third generation, it will become 8. So like that, whatever is the number of generations, so if the number of generation is 20, the number of bacteria will be 2 into 2 raised to the power of 20. If we start with 1, if we are starting with 1 million, then we'll have to consider that. So 1 million into 2 raised to the power of n. That will be your total number of bacteria in the population. So this is clear to everybody what a generation time is. If you have any doubt, please ask. If you have any doubt in this, please let me know. Please write it in the chat box.
No doubt, ma'am.
Okay. What about others? Okay. Moving on to the next question. Question number 3. Which among the following is not a phase in bacterial growth? Which among the following is not a phase in bacterial growth? Option A, stationary phase. Option B, lag phase. Option C, log phase. Option D, dark phase. Which among the following is not a phase in bacterial growth? Okay. So option A is stationary phase, option B is lag phase, option C log phase, and option D is dark phase. What do you think is the answer? Please write your answers in the chat box. I request everyone to write the answers. Okay, I have one answer in the chat box. Option D. Okay, two answers. Both of them are option D. What about others? All of you write the answers in the chat box. If you won't answer, or if you won't tell me if you're understanding or not, how would I know if you're paying attention, or are you getting anything or not? This is a doubt clearing session. If you have a doubt, you have to ask to in order to get it cleared. Okay. Okay, I have 3 answers in the chat box. All of them are D. Let's see the answer. The correct answer is option D, dark phase. So in a bacterial growth curve, so when a bacteria grows, when a bacterial population grows, it follows a curve like this. Okay, so when a population starts growing, uh, it will first try to get adjusted with the medium, with the environment. So we generally grow the bacteria in a medium, in certain medium which has carbon source, nitrogen source, like that. Okay, so whatever is the medium, whatever food source you are giving, since it is new to the cells, it will first adjust itself to the new environment, right? It's like when you if you are visiting a new place which has a very different environmental conditions, climatic conditions than what wherever you are living right now, you will take some time to adjust to that new environment, right? So similarly, the bacteria also will take some time uh, to adjust to the new environment before it can start growing. So that part where it is adjusting to the environment, that is the lag phase. In this phase, there is no cell doubling, but the cell starts preparing itself for doubling. So you remember I was telling you about binary fission, right? So before this division is happening, before this step is happening, all these steps are also happening, right? So the cell is replicating its DNA, increasing in size. So those things are happening in the lag phase. Okay, the first phase, that is lag phase, those things are happening. Basically, cell is preparing itself so that it can enter the log phase or the exponential phase where it can start doubling. Okay, to enter the next phase, it has to prepare itself. So it adjusts itself to the new environment, to the new medium, and then it starts preparing itself. It replicates its DNA, synthesizes a new copy of DNA, replicates it, and then uh, increases its size. Everything is happening in the lag phase. Then once the cell is completely prepared for the next phase, it will enter the next phase, that is the exponential phase, or also known as log phase. Uh, it is a logarithmic phase. So I told you the bacteria divides in an exponential manner. So it will keep on doubling after every generation. So with time, it will linearly increase, but there is a certain point after which the growth becomes static. So after which the population cannot double anymore. Okay. And why does this uh, static uh, phase comes? This phase is called stationary phase. And why does it come? It basically occurs because we have certain we are growing the bacteria in a medium, right? So we have given it certain food to eat. Now there will be a point when the food is going to get over, unless you are continuously adding uh, feed to the culture. There's no way that the food will be unlimited. Okay, so there has to be a point where everything is over and now the bacteria can't grow anymore. So again, there will be a stationary phase. So the stationary phase is there for certain time period before it enters the death phase, where the cells start dying. Now eventually there will be a point where the cells will die. Okay, so in the stationary phase, the cells are neither growing but not nor dying. Okay, so they are neither growing nor dying. Okay, so it is stationary. It is not increasing in number, but it is also not decreasing in number. But in the death phase, cells are actually decreasing in number. So this is showing the number of bacteria in logarithmic scale. So you have the lag phase where the number is not increasing because the cells are preparing itself. And then you have the log phase or the exponential phase where the number is increasing linearly in a logarithmic scale. So it is continuously doubling in number. And then you get the stationary phase where again there's no cell number increase, and then you have the death phase. Okay, so there's no phase called dark phase is there. So it bacterial growth curve has four phases: lag phase, exponential phase, or you can call it log phase, stationary phase, and then death phase. Okay, the growth curve is divided into four different parts. Is that clear to everybody? Anybody has any doubt in the bacterial growth curve, please ask. Is this clear? Okay. Okay. Then I'll move on to the next question. Question number 4. Any element that is least available relative to the requirements of an organism is defined as dash since the growth of the organism dash. So we have to fill in the blanks. Any element that is least available relative to the requirements of an organism is defined as the dash since the growth of the organism dash. So the first option is for the first blank, the word is limiting nutrient, and since the growth of the organism is directly proportionate to the concentration of the limiting nutrient. Second option is the word is toxins, and since the growth of the organism are compromised in the presence of them. Third option is uh, heavy metals, and since the growth of the organism depends on the molecular weight of the metals. And fourth option is maximizing elements, since the growth of the organism is directly proportionate to the presence of the elements. Okay, so what do you think is the correct answer? So any element that is least available relative to the requirements of an organism is defined as the, what do you think is the correct answer? Write your answers in the chat box, everybody. Okay, I have two answers in the chat box. One says option A, one says option B. Okay, what about others? Everybody write your answers in the chat box. We already have two different answers. So what about others? Let's see which answer gets the maximum votes. What do you think is the answer? Is it A, B, C, or D? We already have A and B in the chat box. Nobody wants to answer? Okay, I'll show you the answer. The correct answer is option A. So any element that is least available relative to the requirements of an organism is defined as the limiting nutrient, since the growth of the organism is directly proportionate to the concentration of the limiting nutrient. So basically what happens is, if we are adding, let's say, 4 components to the media for the organism to grow in it, so we have, let's say, A, B, C, and D, 4 components in the media. Okay, now the component B, we are adding it in a smallest quantity. Okay, so that will become the limiting agent. Okay, so once B is, and the organism requires all 4 components to grow. Okay, organism requires all 4 components to grow: A, B, C, and D. Now I have added B in lesser quantity than all the other 3. So naturally, since B is less, it will get over first. Once it gets over, the bacteria is not gonna grow anymore because it cannot consume A, C, and D alone. It needs B. Okay. So if it needs B and B is already over, so the growth will also be limiting. So basically, limiting nutrient is the element that is least available relative to the requirements of an organism. So it is least available to the organism. So it will deplete first. Okay, so that is the limiting nutrient, and it is uh, it actually makes the growth of the organism uh, stop because the nutrient is now over. So basically, the growth of the organism is directly proportional to the concentration of limiting nutrient. So whatever the concentration of limiting nutrient is there, the growth will solely depend on that. If one of the component is in excess, then it doesn't matter. The organism is going to grow its full life cycle because that thing is not gonna get over anytime soon. It is in excess. But something is in limiting quantities, then it is a problem. Then the organism's growth becomes dependent on that component. Okay, is it clear? Everybody understood what limiting nutrient is? If you have any doubt, please ask. This is clear to everybody? Okay, I hope that's clear. Moving on to the next question, question number 5. Identify the true and false statements from the following. First statement is selective media are used for the growth of only selected organism. Second statement is differential media are used to distinguish one microorganism type from another growing on the same medium. So first option says both the first statement is true, second statement is false. Second option says both the statements are true. Third option says first statement is false, second statement is true. Fourth option is for both the statements are false. So what do you think the correct answer is? First statement is selective media are used for growth of only selected microorganisms. Second statement is differential media is used to distinguish from one microorganism type from another on the same medium. Tell me, what do you think is the correct answer? Write your answers in the chat box. All of you write the answers in the chat box. Come on, write your answers in the chat box. Do you think the first statement is true? Selective media are used for growth of only selected microorganisms. Is it true or false? Okay, first statement is true, somebody said. Okay, what about the second statement now? Differential media are used to distinguish one microorganism type from another growing on the same medium. Is it true or false? What do you think about the second statement? Is it true or false? Okay, we have one answer as option B. That is both true. Okay, Dharmika also says true. So both the statements are true. That is the correct answer. So selective media, it is used for growth of only selected microorganism, and differential media are the ones which is able to distinguish between two microorganisms on the same medium. So I'll uh, show you examples. So this is the proper definition. So selective media used for the growth of only selected microorganism, media that inhibit the growth of unwanted microorganisms and support the growth of organism of interest are called selective media. Selective media contain particular ingredient that inhibit the growth of certain microorganisms. So basically, that medium has certain ingredients that are not going to allow certain microorganisms, but will allow only specific microorganisms to grow. Okay. And then differential media, it has components that can allow groups of microorganisms to be visually distinguished by the appearance of the colony or the surrounding media, usually on the basis of biochemical differences between the two groups. So basically, selective media has certain components that is going to inhibit the growth of certain group of microorganisms, but will allow certain other groups to grow. So that will select for that particular group. It will favor one group. Differential media, on the other hand, it will allow both the groups to grow, but it will have certain component that will make one group uh, to be one group will look different on the plate than the other. So you can look at the plate and tell that this bacteria belongs to group A and this bacteria belongs to group B, like that. Okay. So the first example is McConkey's agar. So it is a selective and a differential medium. So you can see here the photo. Uh, this photo you can see. So the plate has both the pink colonies and the yellow colonies, right? And in these photos, you can see E. coli, Enterobacter aerogenes, and then Proteus vulgaris, and then Staph aureus. So you can see Staphylococcus aureus, it is not growing on this plate, but all the other organisms are growing on this plate, right? So the reason why Staphylococcus aureus is not able to grow on this plate is it is a gram-positive bacteria. So McConkey's agar contains uh, it contains the dicrystal violet and it has bile salts. So these ingredients, they inhibit the growth of gram positive bacteria. So they don't allow the gram positive bacteria to grow. So only gram negative bacteria can grow. So that is the selective part of it. So McConkey agar is selective media because it can selectively allow the growth of gram negative bacteria and inhibit the growth of gram positive bacteria. So only one type of bacteria can grow. Okay. Now how it is differential. So differential part comes from the fact that it has lactose and it has a pH indicator neutral red. Okay. So the plate, this agar, McConkey's agar, it can actually differentiate between the lactose fermenting and non-lactose fermenting colonies. So if lactose that is added as the carbon source in the medium, if it is fermented by the bacterium, it will turn the pH of the medium acidic, and because we have the pH indicator that is neutral red, so at a acidic pH, it will become bright pinky red in color. So you can see the lactose fermenting colony gives a bright pink color. You can see here also it gives a bright pink color. Whereas the non-lactose fermenting colonies, they will become colorless or yellowish in color. Okay, so these are non-lactose fermenting colonies and these are lactose fermenting colonies. Okay, so this is McConkey's agar. It is selective as well as differential medium. Anybody has any doubt in this? I have another example for the same thing. This is EMB agar or eosin methylene blue agar. So this is also selective and differential. How it is selective? It is selective because it allows the growth of gram negative bacteria but inhibits the growth of gram positive bacteria, just like McConkey agar. Uh, just like McConkey agar, EMB agar also inhibits the gram positive bacteria because of the presence of methylene blue. In McConkey agar, bile salts and crystal violet was inhibiting the gram positive bacteria. In EMB agar, methylene blue is inhibiting the gram positive bacteria. Okay. Differentiating factor is again lactose fermenter and non-fermenter. Since eosin dye is there, so there are two dyes in this media, eosin and methylene blue. So eosin and methylene blue combines together and produces a color change when lactose or sucrose is fermented. So fermenting lactose fermenting colonies actually produces blue-black colonies with a green metallic sheen due to the amide bonding of the dyes in acidic conditions. Okay, so these two dyes combine, and because of their bonding, we see a green metallic sheen. So you can see here in this figure behind the E. coli colonies. So the colonies are blue or black in color like this, and you can see there is a green metallic sheen around the colonies. Okay, so that shine is there, greenish shine is there, metallic shine is there. So that is because these are lactose fermenting colonies, and this is lactose non-fermenting colonies. Okay, this is clear to everybody. So this is also an example of a selective as well as differential medium. Everybody understood this? Anybody has any doubt? What is selective and what is differential medium and their examples? Clear to everybody, right? Okay, I'll move on to the next question. Match the following. So we have 6 different temperature ranges given, and we have different microbial growth conditions written on the right hand side. So you have to match the left column to the right. So left column we have different temperature ranges. We are in degree Celsius. So -30 to 0 degree Celsius, then 0 to 5 degree Celsius, 5 to 16 degree Celsius, 16 to 52 degree Celsius, 52 to 62 degree Celsius, and 62 to 130 degree Celsius. And on the right hand side, we have first row says refrigerator temperature. Many allow slow growth of spoilage bacteria and very few pathogens. Second row says many bacteria survive, some may grow. Third is temperature in this range destroy most microbes, allowed although lower temperature takes more time. Fourth is very slow bacterial growth. Fifth is rapid growth of bacteria. Some may produce toxins. And sixth is no significant growth below this point. So just look at the options carefully and write your answers in the chat box. What do you think the correct answer would be? Match properly and see. What would be the correct answer? Write your answers in the chat box. Anybody? Read the options carefully, try to answer. If you're not understanding, then also let me know what part you're not able to think so that I can help you with that. No one can answer. Okay, I'll show you the answer then. It is option C. So we'll go one by one. So the first temperature range is -30 to 0. So -30 to 0 degrees Celsius, this is the temperature range where no significant growth can be seen because it is the freezing point of water. Most of the like all the microbes grow in aqueous environment only. So and if the water is going to freeze, it cannot actually grow below 0 degrees Celsius. So -30 to 0 degrees Celsius, nothing will grow, no significant growth will be there. Then comes 0 to 5 degrees Celsius. So 0 to 5 degrees Celsius is the refrigerator temperature. So you know the refrigerator at our homes, that has uh, somewhere between 0 to 5 degrees Celsius of temperature. And there are certain microorganisms that can grow. So you know, even if we keep store our food inside the fridge, a lot of times some fungus or something can grow if it is kept for a very long time. Some microorganisms can grow even if uh, the food is kept inside the refrigerator. So there are certain microorganisms that can have a growth, but it will have a very slow growth. So it's not like something will grow in your food immediately the next day. If you are keeping the food item for long period of time, let's say there's a rotten tomato or very old tomato in your fridge. So that can, there might be a chance you can see white or blackish colonies growing or fungal hyphae growing on the tomato. So those kind of things. So it won't, it will be very slow growth. It's not going to happen like today you are keeping something in the fridge and tomorrow you are getting, you are seeing the bacteria grow, bacterial or the fungal growth, microorganism growth on the uh, food item. Okay. So 0 to 5 is the refrigerator temperature. Okay. Then comes 5 to 16 degrees Celsius. So 5 to 16 degrees Celsius, many bacteria can survive, but there are very few that can grow. So the ones that can grow in 0 to 5, they can definitely grow in 5 to 16. There are some that can grow, but most of the bacteria can survive 5 to 16 degrees Celsius. Uh, and there might be slow growth also in most of the bacteria. Okay. Then comes 16 to 52 degrees Celsius. So this is the temperature range. This is the temperature range where rapid growth of bacteria is seen, and there are certain bacteria that can produce toxins as well. So 16 to 52 is the best growth range for all the all the bacteria and other microorganisms as well. Then comes 52 to 62 degrees Celsius. So at this temperature range, very slow bacterial growth will be there again because we are going from the optimum, we are going higher from the optimum range. And then comes 62 to 130, where most of the microorganisms will be destroyed because it is very high temperature. It cannot survive at such a high temperature, and they will destroy the microorganisms. Okay, this is clear to everybody. Everybody understood? If you have any doubt in any of the temperature ranges and the statements that are associated with them, let me know. If you have any other kind of doubt, also let me know. Okay, then I can move to the next question. So this is just showing how temperature affects the microbial growth. So at a very low temperature, the enzymes won't be active. That's why the growth is slow, and then the growth kind of increases since the enzyme activity increases with temperature, and the enzymes best work at the 37 to 40 degrees Celsius range, and then beyond 50 52 degrees Celsius there's no significant growth. Okay, moving on to the next question now. Bacteria that have an optimum temperature of 35 to 39 degrees Celsius are called: option A, psychrophiles; option B, mesophiles; option C, thermophiles; and option D, psychrotrophs. Bacteria that have an optimum temperature of 35 to 39 degrees Celsius are called. The bacteria that can grow in this temperature range, what are they called? Option A, psychrophiles; option B, mesophiles; option C, thermophiles; and option D, psychrotrophs. Write your answers in the chat box. What do you think would be the correct answer? Is it psychrophiles, mesophiles, thermophiles, or psychrotrophs? Okay, we have one answer, psychrotrophs. What about others? Bacteria that can grow between 35 to 39 degrees Celsius. What are they called? What about others? Write your answers in the chat box. I have one more answer, option B, that is mesophiles. Okay. Anyone else? Anybody else wants to answer? Okay, so I'll share the correct answer. The correct answer is option B, mesophiles. So option B is the correct answer here. So we have psychrophiles, mesophiles, thermophiles, and psychrotrophs, right? So the term philes means loving. Okay. So psychrophiles are the bacteria that can grow at colder temperatures. Okay. So they can grow at about 15 degrees Celsius or lower than that. Okay, so psychrophiles are extremophilic bacteria or archaea, which are cold loving and have a optimal temperature for growth at about 15 degrees Celsius or lower, maximal temperature for growth at about 20 degrees Celsius, and minimum at about 0 degrees Celsius. Okay, so psychrophiles are basically cold loving. Okay, then comes thermophile. So thermophile is like psychrophile is cold loving, thermophile is hot loving. So it can survive at very high temperatures. So uh, we have many hot water springs in India, right? Uh, so in those hot water springs, the water temperature is actually very high. It's generally about 60, 70 degrees Celsius, or sometimes even 100 degrees Celsius. Okay. So at those high temperatures also, if you take the water and just culture it in a on a plate, you will see there are certain microorganisms that can grow at such high temperatures. Okay, so that means there are organisms that are called thermophiles. They can survive. They can grow at a temperature above 40, 41 degrees Celsius, and it can grow up to 122 degrees Celsius. So uh, you can imagine at very very high temperature it can grow. So there are certain places where volcanic eruption happens, right? So in even in those areas, the area around the volcano is very hot, but even in those area there are certain bacteria that can grow. Okay, so those are thermophiles. Then so similarly psychrophiles, you can imagine where it can grow. So if you go to Arctic regions where the temperature is always freezing cold, even at those areas there are certain bacteria that can grow. So those are psychrophiles. Okay, then comes the term psychotrophic. So till now we are dealing with the term phile. So phile, I told you it's love. Phile means love. Okay, so psychrophile is cold loving, thermophile is hot loving, mesophile is something that can that loves the moderate temperature. Okay, but now we have another term called psychotrophic. So when we write the trophic as a suffix, it basically means that it doesn't really grow at the colder temperatures, but it can actually survive. It will have the optimum growth at higher temperature, like at 37 degrees Celsius, but it can also grow slowly at the lower temperatures at 7 degrees Celsius or 14 degrees Celsius, like that. Okay, so it can tolerate the colder temperature, but it will the optimum growth will be at the higher temperatures only. Okay, so these are the four terms. Is it clear to everybody? Let me know if you have a doubt. Okay, moving on to the next question. What is the upper temperature limit for growth of algae and insects? What is the upper temperature limit for the growth of algae and for the growth of insects? So option A is 38 degrees Celsius for algae and 40 to 45 degrees Celsius for insects. Option B is 55 to 60 degrees Celsius for algae and 60 to 62 degrees Celsius for insects. Option C is 45 to 50 degrees Celsius for algae and 55 to 60 degrees Celsius for insects. And option D is 55 to 60 degrees Celsius for algae and 45 to 50 degrees Celsius for insects. Anybody knows the answer for this? This is very straightforward. It doesn't require any logic as such. If anybody knows the answer, you can write it in the chat box. Nobody? I'll tell you the answer to this. The correct answer is option D. So commonly the upper temperature limit for algae is 55 to 60 degrees Celsius, and that for insect is 45 to 50 degrees Celsius. Okay. So moving on to the next question. Moving on to the next question now. Uh, what do you mean screen was clear? Can you see the question number 9? You might be having some network issue. Can anyone else confirm if they are also facing the same problem? Dharmika, you might be having some network issue. You can maybe disconnect and rejoin the call. Others can see my screen, right? Can anyone else confirm if you can see my screen or not? Is it clear now, Dharmika? Okay. Okay, then I'll start with the question number 9. The ratio between the vapor pressure of a food item when in a completely undisturbed balance with the surrounding air media and the vapor pressure of the distilled water under identical condition is called. What is the ratio between the vapor pressure of a food item in a completely undisturbed balance with the surrounding air media and the vapor pressure of the distilled water called? What is the ratio between these two are called? Option A, water activity. Option B, evaporation rate. Option C, moisture content. And option D, saturation point. What would be the correct answer here? Write your answers in the chat box. What do you think would be the correct answer? Ratio between the vapor pressure of a food item when in a completely undisturbed balance with the surrounding air media and the vapor pressure of the distilled water under identical condition is called. I have one option answer as option C, that is moisture content. Okay, what about others? Anyone else wants to answer? Okay, we have two answers now. Both of them are moisture. What about others? Everybody agrees to this answer or anybody wants to answer something different? Okay, I'll show you the correct answer. The correct answer is actually water activity. That is option A. So water activity actually tells us about the moisture content, but it is the ratio is called the water activity, the ratio of the vapor pressure of the food with respect to the vapor pressure of the water. That is called the water activity. So water activity of a food is the ratio between the partial vapor pressure of the food itself when in a completely undisturbed balance with the surrounding air media and the vapor pressure of the distilled water under identical condition. Water activity of 0.80 means the vapor pressure is 80% of pure water. So basically it shows the moisture content inside the food, and it is a important parameter to determine the food safety. So higher the water activity, that means moisture content is high. That means more water would be available for microorganisms. So microorganism growth will be faster in such food items. Whereas if you have lower water activity, that means the moisture content is low. That means the uh, microorganisms cannot grow quickly in that food item. So, all those dehydrated foods you get, uh, the instant noodles and all. So, all those have a very low water activity. They are almost completely dry. So, that's why they can be stored for a longer time. It kind of increases the shelf life of the food. Okay. Lower water activity means it is going to have a longer shelf life. The food item is going to have a longer shelf life. Whereas higher water activity means that it can actually lead the microbial growth can start very quickly in that. Okay, and the water activity increases with temperature. That is natural, because with the increase in temperature, the solubility of certain salts and sugars will also increase in water. So overall water activity also increases. Moisture content of a product can be measured as the equilibrium relative humidity, that is ERH, expressed in percentage, or as water activity expressed as a number ranging from 0 to 1. Okay, this is clear. So water activity is generally ranging from 0 to 1, and if you multiply it with 100, you will get the moisture content. That is, like, here the water activity of 0.8 means 80% of uh, moisture content is there. Okay, is this clear to everybody? Do you understand why it is not the moisture content but the water activity? That is the ratio between the partial vapor pressure of the food with respect to the pure water under the same conditions. Moving on to the next question then. Again, a true or false question. So there are two statements. First statement is bacteriostatic inhibits growth. They do not kill cells. And second statement is bacteriolytic kills bacteria but do not cause cell lysis. So what do you think? The first statement or and the second statement, whether they are true or false? So option A says the first statement is true, second statement is false. Option B says both statements are false. Option C says both statements are true. And option D says first statement is false, second statement is true. What would be the correct answer? Bacteriostatic inhibits growth. They do not kill cell. Bacteriolytic kills bacteria but do not cause cell lysis. What do you think? Both the statements are true, false, or one is true, one is false? Come on, write your answers in the chat box. Anybody? Okay, statement one is true, that is okay. What about statement two? What about statement 2? From the name itself, what do you think? Bacteriostatic. From the name, what do you think it is? It says bacteriostatic. So that means it can inhibit the growth, and it is basically going to make the bacteria static. So it is not killing the cells, but it is not allowing it to grow also. So it won't increase in number nor it will decrease in number. Okay, Dharmika says statement 2 is false. Yeah, that is also the correct answer. So bacteriolytic, by the name itself, you can see bacteriolytic. Okay, so it will lyse the bacterial cells. So it will lyse the bacterial cells. That means it is going to kill. So it is going to kill the cells and cause cell lysis. So it will. So the statement says it do not cause cell lysis. So that is the wrong statement. Okay. So option 1 is the correct answer. Option A is the correct answer. First statement is true. Second statement is false. So bacteriostat. Sorry, just a second. Yeah. So bacteriostatic compounds inhibit growth. They do not kill the cells. So example is refrigeration. So whenever we grow a bacterial cell in a culture and then we keep it in the fridge, it stops the growth, but the number of cells is same. So total number of cells and the total number of viable cells, they remain constant over time. The cells do not die. Okay. Bactericidal, in case of bactericidal, it kills the cells but do not cause cell lysis. So the total number of viable cells will decrease, but total number of cells, like the live cells plus dead cells, that will constitute to the total cells. Okay. And viable cells are just the live cells. So live cells will decrease number of live cells, but total number of cells will be there. So the cells are not lysing, right? Cells are not bursting and disintegrating. So cells are there. They are just dead. They are not going to grow. So you can differentiate between the live and dead cells. So total number of cells will remain same. Viable cells will decrease. And in case of bacteriolytic, since the cells will burst and cells will lyse in the medium, so both the total number of cells and the viable cells both are going to decrease in that case. Okay, in case of bacteriolytic. That's clear? I hope that's clear to everybody. So bacteriostatic, bactericidal and bacteriolytic. There are three types of agents. Moving on to the next question. Which of the following terms refer to an agent that selectively eliminates fungal pathogens from host with minimal toxicity to the host? Option A fungicidal, option B homeostatic, option C bactericidal, option D bacteriostatic, and option E germicidal. What do you think would be the answer? It's very clear from the name. Write your answers in the chat box. Okay. Option A. Yes, that is the correct answer. So from the name itself, you can see fungicidal are the agents that selectively eliminates the fungal pathogens from hosts with minimal toxicity to the host. Okay. So fungi, homeostasis or homeostatic, it means self-regulate. It's a self-regulating process. So our body temperature is 37 degrees Celsius, right? So our body, even if it is cold outside or even if it is very hot outside, our body self-regulates its temperature. So that is a homeostatic process. So homeostasis is any self-regulating process by which an organism tends to maintain stability while adjusting to the conditions that are best for its survival. Okay. And germicidals are any substances that kill germs. So germs is a common term that we use for all the microorganisms combined together. So bacteria, virus, and other microorganisms. Okay, that can cause infection and disease. So all pathogenic organisms are commonly termed as germs. Okay, so any substance that can kill the germs are germicidals. And I already discussed about bactericidal and bacteriostatic, right? Moving on to the next question now. Which of the following is not a characteristic of the autoclave? Which of the following is not the characteristic of the autoclave? Option A, the ability to sterilize solutions with endospores. Option B, the requirement for long exposure time. Option C, the use of moist heat. Option D, the use of high pressures. So I told you autoclave is like a pressure cooker. So there are certain characteristics of the autoclave. One of the following option is not a characteristic of autoclave. What do you think is the correct answer? Option A, the ability to sterilize solutions with endospores. Option B, requirement of long exposure time. Option C, use of moist heat. And option D, use of high pressures. I have one answer in the chat box. Option B. Okay. What about others? Anybody else wants to answer? So the correct answer is actually option A. So requirement of long exposure time is a characteristic of autoclave. We generally do the autoclaving process for 15 to 20 minutes. So we have to expose it for a longer time for the proper sterilization. But the autoclave is not able to sterilize solutions with endospores because endospores are very stable structures. So endospores are dormant structures of certain microorganisms, certain bacteria. So certain bacteria can form endospores so that they can germinate later and grow in the medium after a long time. So it can remain uh, in the medium for a long time basically. So to kill the endospores, it's very difficult. So even autoclaving so autoclave reaches up to 121 degrees Celsius, but that high temperature is not sufficient to kill the endospores. Okay. to destroy the endospores. Moist heat, the use of moist heat, that is a characteristic of autoclave. I told you it's like a pressure cooker. So we use the moist heat, we use water to, and it is basically steam sterilization. And use of high pressures also, I told you, that is a characteristic of autoclave. So option A is the correct answer. This is how a autoclave looks like. So you can see it kind of looks like a pressure cooker, right? So it has a coil to heat the water. So we add the water here. We had another vessel inside which we keep our things. So we can keep our flask or beaker with a solution, whatever you want to sterilize, you can keep it, cover it with a foil, and then put the lid on it. So when you close the whole thing, it looks like this. So it has a pressure gauge. So here you can see this meter. This is a pressure gauge. It shows the pressure. So when the pressure reaches 15 psi, we hold the pressure at that point, and we like basically hold the pressure at 15 psi for 15 to 20 minutes so that everything is uh, heated at 121 degrees Celsius and uh, for 15 to 20 minutes. So that is this. So this is the safety valve, just like the vessel of the pressure cooker. So it releases, so basically it opens after 15 psi. So if the pressure goes beyond 15 psi, it will, the safety valves will open so that it will again reach the 15 psi point, and the pressure will be maintained at 15 psi for 15 to 20 minutes. Okay, if the electric, so before the electric autoclave, we used to have uh, autoclaves that were supposed to be heated by gas ovens, just like we heat our pressure cookers. So if you remove the electric coil from here, you can put it over a gas and gas burner and heat the water. Once the water will heat up, the steam will be filled inside the chamber and the pressure will start increasing. Once the pressure reaches 15 psi, you can hold the pressure by lowering the flame of the burner, or uh, by attaching, by regulating the pressure by safety valve. And then once 15 minutes are completed, you can switch turn off the heat, and uh, your autoclave will be completed. Wait for the pressure to reach to zero again, and then you can open the lid, just like uh, the pressure cooker. When you can't open the hot pressure cooker, right? You have to release the pressure from the vessel, and then you can open the lid of the pressure cooker. It's same like that. Okay, I hope that's clear. Okay, we have another true or false question. So two statements are there again. First statement is UV and X-ray radiations are similar in that they damage DNA and have high penetrating power. Second statement is most disinfectants can be used as antiseptics. Tell me, what do you think about these statements, whether they are true or false? UV and X-ray radiations are similar in that they damage DNA and have high penetrating power. Second statement is most disinfectants can be used as antiseptics. So first option says first is true, second is false. Second option says both are false. Third option says both are true. Fourth option says first is false, second is true. What do you think is the correct answer? Write your answers in the chat box quickly. Anyone? Okay, both false. That is the correct answer. So you are right. So both the statements are actually false. So UV and X-ray radiations, they both can damage DNA, but penetrating power is very different. UV has a lower frequency and longer wavelength than X radiation, uh, due to which UV has a lesser penetrating power than X-ray. Okay, so both X-rays and UV has the potential to damage DNA, but uh, penetrating power of X-ray is much higher than UV radiations. Okay. The second statement was most disinfectants can be used as an antiseptics. That is also false. I told you disinfectants are the agents that can inhibit the vegetative pathogens present on an object. And antiseptics are the ones that can prevent the growth of microorganisms on living tissue. So antiseptics, I gave you the example of the Dettol and Savlon that we use on open wound. And disinfectants, I gave you the example of the floor cleaning solution that we use at home while mopping, right? So disinfectants cannot be used as uh, antiseptics because they can destroy only the vegetative pathogens, whereas antiseptics can inhibit the growth of microorganisms on living tissue. Okay, so it can inhibit the growth of all microorganisms, whereas disinfectants will only remove the vegetative pathogens from there. Okay, that's clear. Anybody has any doubt? Please write your doubts on in the chat box as well. We'll move on to the next question. Which of the following method is bactericidal? So I told you what does bactericidal means. Which of the following method is bactericidal? Option A is deep freezing, option B is lyophilization or freeze drying, option C is membrane filtration, and option D is addition of strong acids. What would be the correct answer? Write your answers in the chat box. What do you think would be the answer? D, maybe D. Okay. What about others? Anybody else? Come on, at least try to answer. Okay, so you are right. Option D is the correct answer, addition of strong acids. So strong acids can destroy the cell without causing cell lysis and are therefore bactericidal. Other three methods mentioned do not kill the cells. So deep freezing and freeze drying are processes which are bacteriostatic. So basically it will stop the growth of the bacteria but will not kill the bacterial cells. Membrane filtration is a sterilization process or a disinfection method. So it can basically remove the microbes from a solution. Okay. So addition of acid can actually kill the cells. So that is bactericidal. Deep freezing or freeze drying, these are both bacteriostatic methods. That's clear? Okay. Let's move to the next question. If you have any doubt, please let me know. I hope that's clear to everybody, right? I'll move to the next question then. Question number 15. Antimicrobial activities can be measured using option A, spread plate method, option B, pore plate method, option C, disk diffusion method, and option D, membrane filtration method. Antimicrobial activities can be measured using option A, spread plate method, option B, pore plate method, option C, disk diffusion method, and option D, membrane filtration method. What method can be used to measure the antimicrobial activity? What do you think would be the answer? I think B. Okay, pore plate method. Anybody else wants to answer? What about others? Only one person is answering. How can you measure the antimicrobial activity? Okay, let's see the answer. The correct answer is disk diffusion method, option C. So basically, we use this method to check the antimicrobial activity of certain compounds. So let's say we are checking the antimicrobial activity of the antibiotics, or we are checking the antimicrobial activity of a certain oil that we have extracted from a plant. Okay, just giving a random example. So what we do, we use the small paper disc, we soak it in the solution, and we keep it over the plate on which we have already spread the bacterial culture. So you can see, when we incubate this plate, what will happen, the bacterial culture will grow to all the places, but it won't grow in the area where the disc is kept. So the from this paper disc, since it was soaked in the uh, solution, uh, the solution will now seep into the medium in a radial manner. Since the disc is circular, it will seep into the solution in a radial manner, and depending on the antimicrobial power, so you can see here this antibiotic has higher antimicrobial power than these two, so it is making a larger zone of inhibition, and these are showing smaller zone of inhibition. Okay, so and depending on the diameter of the zone of inhibition, you can determine which antibiotic is best for your for killing your microbe of interest, bacteria of interest. Okay, so this is disk diffusion method. Is this clear? So the other methods that are written here, spread plate method and pore plate method, we use these to count the number of colonies, to basically determine the number of bacterial cells in a particular culture. So we do a CFU counting by doing serial dilution, and we can use either of these methods to plate the cells. Then we have membrane filtration method. I told you it's a sterilization process. Is this clear? What is this diffusion method? Okay. Moving on to the next question then. Decimal reduction time is the time in which decimal reduction time is the time in minutes in which dash of the population at a given temperature will be killed. Option A is 75%, Option B 10%, option C 100%, and option D 90%. Decimal reduction time is the time in minutes in which dash of the population at a given temperature will be killed. What do you think would be the correct answer? Write your answers in the chat box. Okay. I have one answer, option B, that is 10%. Okay. What about others? Pay attention to the question. Decimal reduction time is the time in minutes in which dash of the population at a given temperature will be killed. Okay. So you have to tell the percentage of population that will be killed, not the percentage of population that will survive. Hmm. Okay, I have another answer, option A. Anyone else wants to answer? So I have two answers right now, 10% and 75%. Anybody else wants to answer? Dharmika says 90%. So I have three answers now, 10%, 75%, and 90%. That's all. Anyone else wants to answer? So the correct answer is option D, 90%. So decimal reduction time is basically the time required to kill 90% of the microbial population exposed to specific temperature, or the time at which 10% of the original population of a given organism uh, is remaining. So basically, either 90%, either you calculate the number of organisms that are killed and then see whether it is 90% of the total population, or you can see how many cells are living and you can see if it is 10% of the original population. So the time required to reach that 90% of the killed microbes or the 10% of the surviving microbes, that is the decimal reduction time or the D value. Okay, it is called the D value. Is that clear? Any doubt in this? So the same question can come in another way, that decimal reduction time is the time in minutes in which dash of the population at a given temperature will uh, be living. Okay, so in that case, the answer would be 10%. So you have to read the question carefully. Okay, if that's clear, we can move on to the next question. All of the following methods are used for food preservation except option A, commercial canning, option B, adding sugar, option C, vacuum packaging, and option D, pasteurization. All of the methods are used for food preservation except. So which of the following is not used for food preservation? Commercial canning, adding sugar, vacuum packaging, pasteurization. What would be the correct answer? Write your answers fast. Option D says pasteurization. Okay. You don't think pasteurization can preserve food? I explained what pasteurization is. Okay, I have another answer. Option A, commercial canning. So you don't think canning can actually preserve food? Anybody else? Option C. So all of you gave all the wrong answers. The actual correct answer is option B, adding sugar. Adding sugar is not a way to preserve food. Adding salt can actually prevent microbial growth and it can help in preserving food. But addition of sugar can only uh, improve the texture of the food, color of the food, but it cannot preserve the food from microbial growth because microbes can easily eat the sugar and grow happily. Commercial canning is a process of food preservation. So you see certain canned foods, right? Like you get corns uh, in a can, or you get certain sauces in the can. So those tin cans, they are made from multiple layers of metal, and they are made in such a way that microbes cannot grow inside that can. Okay, microbes cannot enter the can. Then vacuum packaging, that is also very effective method of food preservation, because if you remove all the air, if you pack something under vacuum, it will not have any amount of oxygen or any amount of air to for the for the microbes to grow. Okay, so vacuum packaging, it's a very good method of food preservation. So you might have seen certain food packs that are packaged under vacuum. When you cut, the air fills in and the packet puffs up. Okay, mostly you can see the non-vegetarian food are packaged like that. The packaged non-vegetarian foods are sold under vacuum packaging. Okay, and pasteurization, I told you pasteurization is a process. It's a very commonly used process in milk industry and dairy industry, where the milk is pasteurized to prevent the growth of microorganisms. So that is the first thing I explained. It is a very effective preservation technique. Okay, so the correct answer is addition of sugar. It helps in preserving the color, texture, and flavor, but it does not inhibit the microbial activity. That's clear. Okay. Moving on to the next question. Which of the following is suitable for use on tissues or microbial control to prevent infection? We have already discussed this. Which of the following is suitable for use on tissues or for microbial control to prevent infection? What do we use on tissues? What do we use on living tissues to control the microbial growth? Yes, option B, antiseptic. We use antiseptic. I've already discussed this, so we won't spend time on this one. Okay. Filters having pore size of dash are often used for sterilization. Filters having a pore size of dash are often used for sterilization. Option A, 0.08 microns. Option B, 0.45 microns. Option C, 0.22 microns. And option D, 0.03 microns. Filters having a pore size of dash are often used for sterilization. 0.08, 0.45, 0.22 or 0.03 micron. What should be the pore size of filters for sterilization? Think of it in a way that the pore size has to be smaller than whatever you want to remove, so that it won't pass through the filter, it will be stuck over the filter paper. What should be the pore size? Okay. Naveen says option C, that is 0.22 micron. I have another option. Option A, 0.08 micron. Okay. Anyone else? So the correct answer is 0.22 micron. So bacterial size ranges from 0.3 to 0.5 microns. So pore size of 0.22 micrometer is used for sterilization. So if the pore size is 0.22, bacteria cannot pass through that and we can use it for sterilizing process. Okay, that's clear. Okay, moving on to the last question of this session. Which of the following category of oxygen tolerance is depicted in tube 3? Okay, tube 3. So you have to see in this tube which of the following category of oxygen tolerance is depicted. Option A, aerobic. Option B, facultative aerobes. Option C, anaerobic. Option D, microaerophilic. Is it aerobic, facultative aerobes, anaerobic, or microaerophilic? You have to see the third tube. Come on, tell me the answer. What do you think is the correct answer? Write your answers in the chat box. Option D, microaerophilic. Okay. Anyone else wants to answer? Okay. And another answer is option B, facultative aerobes. Okay, let's see the correct answer. The correct answer is option B. You are right, facultative aerobes. So, the first, so these are basically the tubes are open. So, the oxygen cannot penetrate, it cannot diffuse to longer distance. So, oxygen will penetrate only at the top part. So, the first tube depicts aerobic growth, obligate aerobes that can only grow in the presence of oxygen. So, since oxygen will diffuse only to the top part of the surface of the solution, so the obligate aerobes will grow closer to the surface. Then anaerobes, they will grow since they are sensitive to oxygen, they will grow away from the surface, so they will grow at the very bottom of the tube. Then facultative aerobes, they can grow in either the presence or absence of oxygen. So that's why they are growing throughout the tube. However, the growth is better near the surface. So you can see near the surface, the concentration is high, but throughout the tube, the growth is there. Okay, so they can grow better near the surface since they can respire. Okay. Then comes microaerophilic that can grow at a lower oxygen concentration. So they will grow a little away from the surface. So obligate aerobes are growing very close to the surface. Microaerophilics are growing below that. Okay. And then aerotolerant aerobes, that is the fifth tube, that is showing the growth all over the tube like the facultative aerobes, but uh, they cannot grow better at the oxic zone because they can only ferment. Okay, so they cannot survive very nicely in the presence of oxygen. So you can see the difference between the third and the fifth tube is the growth is throughout the tube, but the growth at the surface is higher for the third tube, but for the fifth tube, it's not the same case. Okay, so third tube shows facultative aerobes. This is clear to everybody? So this is the last question of this session. So let me know if you have any doubt. So if there's no doubt, I can close this session. Thank you all for joining. I'll see you next time. Thank you. Bye bye.